We have a very special guest, Dr. Seema Lakdawala from Emory University School of Medicine. Today, she will give a talk on the topic Understanding of the Leaks of Cattle H5N1. Dr. Lakdawala received her PhD from University of California, and she was a research fellow at Emerging Respiratory Viruses Section, Laboratory of Infectious Diseases, NIH. Before joining Emory, she was a principal investigator at Pittsburgh School of Medicine. Her main research focus is on transmission of respiratory viruses. Seema has so many publications. I can't read all of it today, but many of her good work published in leading journals such as Nature Communication, Post-Pathogen Nucleic Acid Research, and Journal of Infectious Diseases. Please join me in welcoming Dr. Seema Lakdawala. And if you have a question, you can put on the chat box, or at the end of Seema talks, you can raise your hand and I will let you ask the question. Seema, the floor is yours now, please.
Thank you so much, Kobe. You forgot to mention that we were postdocs together, so it's such an honor to be invited to come and speak to the Thai Virology Association. So thank you so much for having me. I will share my screen. Give me 1 second here. Sorry, I had it all ready to go, and then did that work?
Yeah,
Do you
it's work.
see? Okay, great. Do you see the slide now? Perfect. Wonderful. So today about H5N1 and how it's really spreading birds, how it's coming into cows and into humans, and sort of what some of the risks are and where this virus has really come from and perhaps where it's going, unfortunately. So just a bit of background for those who are not influenza experts. Influenza is part of the family of orthomyxoviruses. There are 4 types, A, B, C, and D. Maybe I can make my laser pointer here instead. And there's a lipid envelope. There are 8 RNA genome segments. These are negative sense RNA segments that encode up to at least 11 proteins, but they encode so many important ones that are on the surface of the lipid here, the hemagglutinin in blue, the neuraminidase here in red, and then the matrix protein sort of shown here in green. The hemagglutinin, there are 19 known subtypes, so there's a lot of diversity, though H1s through H19s. You may be familiar with an H1N1 subtype. That is a hemagglutinin of subtype 1 with a neuraminidase of subtype 1. There are 11 known subtypes of neuraminidase, so you can imagine having any variation of HA from 1 to 19 to NA from 1 to 11. That sort of leads to a lot of genetic diversity in nature and amongst all the different animals that are infected with influenza. So here I actually modified this slide recently to include cattle, but influenza viruses are known to infect many hosts, and this is just a small subset, right? So we believe one of the primary reservoirs for influenza is waterfowl. Viruses from waterfowl, such as migratory birds and ducks, can infect humans, pigs, cattle now, we'll talk about that today, aquatic mammals like seals and whales, horses, dogs, cats are another one that I have neglected to put on here, as well as poultry, such as chickens and turkeys. And of course, also bats are considered to be a secondary reservoir. Importantly, humans are known to become infected and cross-transmit the virus back to pigs. So the swine virus that sort of created the 2009 pandemic H1N1 virus, really transmitted from pigs into humans and has gone backwards from humans back into pigs. Ferrets are also a great model system to study influenza viruses because they are naturally susceptible to human influenza viruses. And there is actually also some evidence now, of course, cattle transmission into humans, although sporadically, the same with poultry. So some of the dynamics of influenza virus circulation in humans, we have a long history with influenza viruses. Of course, this human seasonal H1N1 viruses emerged prior to 1918, but really, we think that the Spanish flu or the 1918 pandemic sort of emerged and circulated in the human population. This sort of is shown how it changes across the human population by changes in the color of the arrow, so suggesting antigenic drift, up until 1957, when there was an outbreak of an H2N2, the H2N2 pandemic. And that circulated in the human population until 1968, where there was an H3N2 pandemic. And there was reemergence in 1977 of this 1957 strain, likely from a vaccine trial. And that circulated in 2009. So there was co-circulation of H1 and H3 from during this time until 2009, we have an H1N1 pandemic. And this is called variant because it emerged from swine and it was a similar H1N1 subtype. So it's H1N1 variant. Of course, all of these viruses sort of evolve in the human population. So this antigenic drift, again, is shown by the changes in the color of the arrows as it circulates. But antigenic shift can really be referred to as changes in the hemagglutinin. And so these sort of pandemic shifts are similar to are actually considered antigenic shifts. Of course, what we really don't know is what's happening next. And this is, we're going to talk about today is H5N1. So just as a bit of background about avian influenza viruses, so, bird flu has been with us for a long time. It was first identified in 1959 in chickens in Scotland. There are two different types of avian influenza viruses. There's what's called a low pathogenic avian influenza virus, this is LPAI, or highly pathogenic avian influenza viruses, or HPAI. And all of this is sort of derived from changes in this multi-basic cleavage site here. And just zooming in, the shorter loop here in yellow is the LPAI, and the black loop here in the larger loop here in black is the HPAI. And this is just a surface rendering of the hemagglutinin monomer. So in chickens, highly pathogenic avian influenza viruses are highly lethal. And that's really where they get their name from. So HPAI does not refer to necessarily pathogenesis in humans, but really pathogenesis in avian species. And so in these studies, there are a couple of studies done that really demonstrated that in chickens, HPAI could lead to lethality and severe mortality in avian species, some succumbing to the disease within 2 days to 4 to 5 days, depending upon the strain. But this, of course, is a huge economic burden for poultry farmers, for backyard poultry, and for global economic health, thinking about eggs and all sorts of downstream effects in terms of economic potential. The safety and biosecurity of chickens is paramount. So the first human infection with H5N1 really happened in 1997 in Hong Kong. There were 18 H5N1 cases, human cases in Hong Kong at that point in time, and 7 deaths. And these were related to the Goose Guangdong lineage of 1996. And this was a reassortant event. So what happens, because flu has these segmented 8 segments like I showed you in the beginning, if two different viruses infect a human or infect the same cell, those 8 segments can actually intermingle and viruses. And so here, what happened for this H5N1, there is a reassortment event of H9N2, H5N1, and H6N1. So again, a wide variety of H's and N's, and these came to create this 1997 Goose Guangdong lineage H5N1 that transmitted from quail, sort of like migratory birds, into poultry, this is a chicken, and then sort of made it into humans. And there was a couple of cases that related to this, sort of understanding the disease severity and clinical features. And of course, this really had a huge impact in our risk assessment of H5N1 viruses. In 2003, there was a huge outbreak of H5N1 that sort of occurred all over mainland China, including places of Malaysia and Indonesia. And, there were over 100 cases by 2005, and about 30% mortality. We, of course, as a nation and as a world, we're really worried about the threat of avian flu, given the high mortality and the clear persistence of this virus in these human populations and really at these animal-human interfaces. that was happening. So in red here are the outbreaks with countries with animal outbreaks, and in yellow are sort of ones that were more since 2003 versus 2005. And so a big important thing that sort of really highlighted the issues and sort of the risk of this virus is that these migratory birds that can become infected with H5N1, which are not that lethal in these in migratory birds, right? It's really lethal in poultry, can transmit the virus large distances through flyways, right? So birds originating in places like Thailand or Laos or Vietnam can really, go all the way up through to Russia and Europe and Africa and the US. And so there's lots of different predominant flyways for birds that can transmit this virus across other migratory populations of birds and into what we have seen recently, an expansion of avian species that are infected with H5N1, including songbirds, raptors, condors, etc. And so the current spread of the 2344B clade of H5N1 is sort of like highlighted here, right? There are lots of countries. The only exception right now that we know of is Australia, that has a large surveillance infrastructure in place, but most countries have reported some cases of H5N1, either in birds or in other sorts of small scavenger mammals, as well as in Europe, a large predominance in Europe. In Africa, along the coast of Chile, there's been a lot, as well as in Brazil and all over across the US. And so I'm going to focus a little bit more on the US now, because I really want to now highlight what's been happening currently in our current situation in the US with H5N1. So here I'm just showing you the wide variation of birds that have been infected or found to be infected in the US. This includes commercial poultry in red, backyard birds in yellow, wild birds in green, and wild mammals. And I'll go here in a minute, but what we're finding is that the diversity of birds that are being infected is really wide in the US. There's been a small reassortment event that's happened. And so we really think that that's driving a lot of these adaptations and success in the variety of bird species that are being captured. And in many cases, it is lethal, but in some, it's not. What we're finding, though, is a lot of animal-to-animal spillover. So especially scavenger mammals, such as cats, either big cats or domestic cats, foxes, bears, coyotes. This is sort of just showing the diversity of the different and locations of the different small mammals that are being found to be infected with H5N1 from just 2022 to present. I just pulled this from the USDA this morning. And so I want to just now shift gears and talk a little bit about what's happening in the cattle and what we think is going on and some of the risk associated. And so I'm not sure of how the dairy industry works in Thailand, so I'm really just going to focus this in on what's happening in the US, obviously. So this is a timeline that may be actually a little bit out of date now, but in March 25th of 2024, there was the first herd that was identified as being H5N1 positive. We do believe, of course, from molecular clock analysis, as well as epi reports, that perhaps this introduction happened a few months prior. And I can explain what, so what happened in this March case was that there was an investigation of a lot of what's called mastitis in milk and cows, where the milk is chunky, and it's normally caused by different bacterial species. But there was no clear bacterial pathogen that was creating this after some testing. And so some veterinarians in Texas noticed that there were dead poultry as well as cats with neurological symptoms on these farms, and then tested the animals for H5N1 based on that sort of epidemiological assessment. And then they turned out to be H5N1 positive. And we'll talk a little bit more about where the virus is replicating in cows. By April 1st, we had our first human infection case that was reported. This individual was in Texas, and they reported with conjunctivitis, which means sort of pink eye. And then a number of different states sort of came up. There was a federal order put in a month after the first introduction. having mandatory testing of dairy cattle that were being moved across state lines. And this is actually only for lactating cows. May 22nd and May 30th, there were the two other additional infections. Both had some form of conjunctivitis. This third case here did have some respiratory symptoms, and both of them were identified in Michigan. And so this is sort of just highlighting that clearly there's a lot of cow-to-cow transmission. We think a lot of the cows originated in Texas, and then the virus spread through cattle transport. Unfortunately, there's not a lot of information about cattle transport in the US because it is a commercial industry, and so the government doesn't have a lot of say or a lot of oversight in the movement of these animals. But currently, based on some earlier reports from the USDA, we have at least over 120 herds that are infected in about 12 states, and we have 3 confirmed human cases. And so where is the virus replicating in cows? It's predominantly being seen in lactating cows. Again, the initial reports were from mastitis, as well as cows dropping in milk production. And so what we found from a couple of more recent experimental studies that have been performed by the USDA is that infected animals that have been infected through the intermammary route tend to produce less milk, and that milk does become looking like mastitis or just chunky and yogurty, sort of curdled milk. But here is the first case report of from these veterinarians in Texas. And these were 4 cows. And what I'm showing you here is that they looked at the PCR CT values in milk. And what I just hope you can see here is that the CT values in milk for these animals were in the teens for H5N1, which is really low. There was also in this animal, there was a lot of it was not detected. It wasn't looked at here, but it was found at low CT values in the mammary gland. In a subsequent report by Yoshi Kaoka's group, it was also reported that these animals that were from New Mexico, so these are Texas cattle, these are New Mexican cattle, these are the CT values in the milk. And then they also did plaque assay. So now we're looking at not just CT values, but infectious virus titers. And you can see it ranges from anywhere from 10 to the 2 to 10 to the 7. And this is a lot of infectious virus. So, many of these specimens can have viral loads ranging to 500 to 60 million PFU per mil, right? And when you milk a cow, every milking session, you're getting about 40 gallons of milk. And so that's a lot of milk. And so, and these cows and these farms have like thousands of cows, and they get milked twice a day. So you just imagine the volume of infectious virus contaminated milk. So we were really concerned about this milking process and about how much virus is found in the milk. And so I just want to talk about how this process works in the US. So here is sort of a picture that I grabbed from this Dairy Global that sort of shows the equipment. This is a cow udder right here, and this is a dairy farm worker. What they first do is they actually do a disinfection of all the teats. So they clean all the teats when these animals come in. So they're backed in, and so this is all at eye height. So these workers can work quickly. And then they forestrip, which is just like starting the flow of milk from these udders, from these different teats. And then the letdown of the teat just starts to spray milk onto these floors. So there's a lot of splash potential of milk just falling onto the surfaces here, as well as aerosols being generated. This instrument here is called the claw or the cluster, which is a sort of similar to a breast pump, where it is attached onto each teat, and it has both positive and negative pressure. And so it's collecting the milk from each of the teats, and it has a regulator right here that is measuring the flow rate of the milk that is coming out of each of these different teats of the cow. And then once it hits a certain rate, you see this chain here, this claw automatically releases and swings out. And because there's so much milk during this process, while the cow is cleaned, there is a lot of residual milk all in these cups. And so this this is called the inflation liner, is the black, and the stainless steel are the main components of this claw milking equipment. And again, we thought that because the cow itself is clean, both pre and post milking. However, the milking equipment is not clean. And it has been known that bacterial infections can transmit from cow to cow through contaminated milk. And so we really also were worried about this as a potential for cattle to cattle transmission as well as cattle to human transmission. And so what we wanted to do is assess the persistence of viruses on dairy equipment, so particularly milking equipment. So here's again that timeline. And I've picked 3 states here that we picked around the times that the first cases were identified in those states, either in New Mexico, in Texas, or in Michigan. And around the time of the first introduction, here I'm showing you the variation between the relative humidity in these states, in these specific towns here in these states. It won't really mean much to you all, but the idea here is that most of these barns are open air. And so the environmental conditions in which these viruses are being expelled into are similar to the outdoor air environment. And so here is Amarillo, Texas. These are the different 7 days around the first incidence of the case. And this is in Michigan, and this is in New Mexico. And so we really picked 70% humidity as a humidity range that was really capturing a lot of what was happening. Of course, we've gone through and now done a few more, but we thought 70% was really critical, especially for Amarillo, Texas, around the first case. And so what we did was we took H5N1, this is the cattle flu virus. We diluted it 1 part to 9 parts in raw unpasteurized milk, or in PBS. And then we made these small 1 microliter droplets onto either inflation lining. So this is this sort of rubber lining that we cut into squares, or stainless steel. And then we collected a time 0 in 500 microliters for an immediate recovery so that we can see where our starting point was. And then we aged the droplets at 70% humidity for 1 hour, and then collected the droplets to look at the change in viral titer. And so here I'll walk you through the data. This is again a cattle H5 virus. I've written it out here. This data was also just recently published in the Emerging Infectious Diseases journal from CDC. So here on the inflation liner, PBS is first. You see that at time 0, there's quite a bit of PBS. 10 to the 4 was our starting point, roughly. And then there was a huge log. It was undetectable after 1 hour. However, in raw unpasteurized milk, the virus is now highly stable, and this decay, what's called our log decay from time 0 to 1 hour, is actually very modest. The same is true on stainless steel. You see that the virus will decay 100% at 70% humidity after 1 hour, but it is protected in the presence of milk. We believe it's because of all of the proteinaceous material found in milk. We, of course, wanted to do additional studies, and we needed a good surrogate. So we compared this to the 2009 H1N1 pandemic, also in raw milk, for 70% humidity for 1 hour. And here, what I'm going to show you now is log decay, which is the titer at time 0 over the titer at time 1. So as virus titer goes down, log decay goes up. So inflation liner has very little decay because the titers were quite high, both for cattle H5 shown in blue and the 2009 pandemic H1N1 virus shown in tan. And that's also true for stainless steel. The log decay is modest, right? It's a little bit higher here, but it's still under a log decay of 2, which is sort of our threshold for a robust decay. We then used the 2009 pandemic virus to actually look at a couple of things that are really challenging to do in BSL-3 with a select agent. We looked at the decay over time on both inflation liner and on stainless steel. So now here again is virus titer. You see over time, from time 0 out to time 5, you're still detecting infectious virus even after 5 hours on inflation liner at 70% humidity. However, by 5 hours on stainless steel, most of the virus has been decayed. And what this tells us is that the virus is highly stable on this equipment. And so this equipment really can serve as a potential fomite contamination source in transmission. And given the high viral loads and the susceptibility in the mammary tissue, it seems like a likely culprit. We then wanted to look beyond just 70% humidity. So we looked at 50% humidity and 30% humidity. This is sort of plotting that same data that I showed you earlier, just a different way. Now, instead of looking at the variation, we're just looking at the median. And instead of 3 states, I'm showing you here now 6. And so, we picked these 3 different humidities to sort of capture the variation amongst the different humidity conditions. We then looked at inflation liner in blue, stainless steel in black, and copper in this coppery color. And that's because copper can really be an antiviral. And so it tends to inactivate virus quickly. And so what you'll see is that at 70% humidity, most of the time, copper has completely inactivated the virus. Although at lower humidities, it's stable. At all humidities, the virus is stable on inflation liners. So that's really concerning given all of the different places geographically that this process happens. We then wanted to understand the persistence of these viruses and potentially aerosols that are generated during the milking equipment, during the milking process. And so we took these dilutions of the 2009 pandemic H1N1 virus in unpasteurized milk. We put them into a medical nebulizer. We aerosolized the virus. So medical nebulizers create small aerosols, about 2 to 4 microns in size. We then collected on an open-face PTFE membrane filter that was pulling at about 5 liters per minute, which is similar to our just sort of tidal breathing rate. Pre and post nebulization, there isn't a lot of loss in titer, as to be expected. And then if we collected for 30 seconds or 5 minutes, we definitely saw recovery of infectious virus, such that aerosolized viruses remain infectious. But these are, of course, short titles, and this is directly on top of it. So we have, we're looking at this a little bit further, but one of the things we did initially was use a 30-second nebulization, but move the filter cassette either 3 inches away, 6 inches away, or greater than 6 inches away, and really showed that the further away you are, the less likely you are to sort of breathe in infectious virus, at least at the doses we were looking at here, which are on par with what's been reported to have be in the milk and literature. And so this is really important, but as I mentioned before, these animals have to be milked, and there's a lot of milk that's produced from a single milking session of a single animal. And so even when these animals have mastitis or chunky milk, it still has to be collected. And so these animals are still connected onto a separate tank. So these don't go into the bulk larger tank that all of the cows go into, but into a smaller one that's just holds this unit. And the unfortunate thing is that a lot of these will then just be dumped down into like the manure lagoons or onto the fields. And we think that this is responsible for a lot of spillover events or potential transmission events to cats. It also then, of course, in the bulk tanks, even when these cows are milked, if they're not chunky, and which tends to be that we're definitely finding virus in milk that looks normal, they go into the consumer population into plastic jugs. And so we looked at the persistence of viruses in bulk solution in these different materials, both either in plastic or stainless steel. This is either at 70% humidity or at 4 degrees. And you see roughly here, this is again log decay now, not virus titer, but there is very little decay at any of these time points, either out to 24 hours or here in 4 degrees, we went out to almost 3 days. And so our conclusions are that cattle H5 viruses are really stable in pasteurized milk. They can remain infectious for up to an hour. So there's a real concern for cattle-to-cattle transmission, where cattle are milked continuously every 10 minutes. A new animal is being milked. And so aerosolized viruses also in this process could be infecting humans that are near the source for where the aerosols are being produced. And milking equipment or aerosols that are generated can drive transmission. Contaminated milking equipment or aerosols could drive transmission between cattle and humans, as well as we definitely think between cattle for the milking equipment. And with that, I'll thank my team. A lot of the work done in BSL-3, all the work done in BSL-3 was done by Valerie Lesage at the University of Pittsburgh, who is holding down my fort in Pittsburgh. We run a joint team now up there. And then I have our team down in Atlanta, and AJ Campbell did all of the H1N1 persistence studies. And we have great collaborators both in the University of Pittsburgh and Virginia Tech on aerosol science, and Florian provided the H5 introductory slide. So I appreciate his contribution. Thank you.
All right, thank you very much, Seema. Great talk as always. Have a quick question for you before the audience start to ask the question. Seema, do you know the receptor uses of this virus? Is this the
Yeah,
same thing? Yeah, okay, go ahead.
it is definitely a 2-3 preference. It is
Okay.
not the receptor preference is still highly 2-3. There are some reports that perhaps it's binding to longer chains of 2,3. Again, it's, sort of the complex glycan structures. But there are some, issues with really teasing apart the glycans beyond that because of the limitations of the microarray. But it is definitely a 2,3 preference.
Okay, so the question from the chat box, do you have any information on the source of the infection in cow? Are there any bird nearby?
Yeah, so there are a lot of birds on these farms. Again, these barns are open air. So the dairy farms that I have visited, you see birds all over. One like pigeons flying in. Sometimes they'll have poultry on farms, not often. But you definitely have birds eating the feed that the cows eat, right? There's a lot of different types of feed. So that in the barn areas, there's birds with the cows there, as well as in these open silos where they store the feed, birds are there. It's a really important question about how did this introduction happen? We actually currently, based on the genetic information that we have from the sequences, think it was a single introduction, which seems really bizarre because it definitely seems that they're highly susceptible. And so it may be not that it's the feed and like a respiratory inoculation, but more that perhaps there was a barn in Texas where a bird was licking the milk on the milking equipment, and somehow so we really think it's an intramammary inoculation. That perhaps that event doesn't happen as often. It's
Okay,
my guess.
okay. Seema, this is going to be a very difficult question for you. What is the solution for the farmer?
Yeah, it is actually a really hard question. And I will say that what I've been suggesting, there's two things that are really important here. One is that we have to protect the dairy workers. And I think, these cows have to be milked. This dairy industry is a $250 billion industry in the US. It is a really important economic consideration here. And so we can't just say like, kill all the cows. That's not the right solution. But what we can do is say for workers, a big protection, I think, would just being wearing a face shield, right? It's hot and steamy in these environments. But a face shield, I think, will be really protective because we think they're getting hit in their eyes. That's causing the conjunctivitis. There's a lot of spill and splatter, and probably the high viral loads in that dose that they're getting is what's initiating an infection. A larger problem is actually how do you prevent cattle to cattle transmission? Because it's really hard to clean this milking equipment. Nobody wants to get any sort of disinfectants in like a milk supply, right? That's a huge no-no. And so I've talked to a couple of people, and I'm really trying to find farmers to figure out like what can we do to sort of prevent the virus from remaining infectious on these cups and then being reused.
Mhm.
Um, it's hard, 100%.
But Seema, since it is kind of like a droplet, right, in the environment nearby, did you get a chance to look? Are there any contaminate nearby the farm?
Yeah, so definitely we think that there's a lot of contaminated equipment within the farms itself, right? Because I mean, it was amazing to me. I went to probably one of the cleanest farms because it was a university farm, and it there was, I mean, the milk is just spewing everywhere, all over the cement floors. Like I got splattered and I was standing far away. Like the equipment is dirty. They're peeing, they're pooping. They're like, and it's like every 10 minutes there's a new set of like 30 cows just coming in and doing the same thing. And these people are wearing gum boots and aprons and gloves, but there's no face protection. And so they're just working as fast as they can to get all of these cows milked, so that they can then shut everything down and clean it and do the next set. And so it is, I think there's contamination everywhere. There's a lot of wastewater surveillance happening now near farms, trying to find like backwashes and we're definitely finding that as well.
Okay, one next question, Seema. Human to human transmission from Dr. Samaporn, I think. Do you think seem like human is a dead end host for the virus, or you think it's possible for human to human transmission?
I think, of course, I think human to human transmission is possible. I think right now the virus is not well adapted to humans. There's a couple of really silver linings, I guess, to see right now. We've had 3 cases and all have been presented fairly mild and all have recovered with oseltamivir treatment. not that it was even like, required. They weren't severely ill. Um, and I do think that perhaps whatever infections we are missing are probably mild as well. I do think we're missing some infections just given how widespread this is. My concern always is that as these viruses replicate in humans, right? These are RNA viruses. They have error-prone polymerases. They like to change. And so it only takes like maybe someone who's immunocompromised or who lives in a very close community, to sneeze or to have just like, a few of the right mutations, and we could potentially have more transmission events. And that's the concern always. So really protecting the workers from getting infected, I think, is a critical piece, as well as blocking cattle to cattle transmission.
Demand any chance that you know the viral titer in infected people from the farm?
Yeah. Yeah. So there it's been challenging to at least my understanding from the CDC is that it's been challenging to grow a culture, the infectious virus. The Most of the CT values have been in the mid 20s to 30s. The ocular swabs tend to have more virus than the nasal swabs. So they're doing both nasopharyngeal and ocular swabs. However, that makes a lot of sense because the two first cases had had no respiratory presentation.
Okay. So two more questions in the chat box, Seema. What is the purple sample for high part surveillance in dairy cattle?
Yeah, so this is really good. I think one of the most important things to do is look at the milk. For lactating cows, if you can get a milk sample from every teat. So what we're also finding is that there isn't dissemination from one teat to another. So some teats will be positive, but other teats won't. I think that's probably the best sample to get to look for H5 in these animals. We're not there's really low involvement in the any part of the respiratory tract right now, at least epidemiologically. And so that would make the most sense.
Target cell, Seema, what is the target cell?
Yeah. So we're looking, a lot of people are looking at this, but definitely the
Uh-huh.
epithelial cells in the mammary gland are antigen positive. And it is really fascinating. I don't know much about the mammary tissues, but there's a lot of work being done right now to try to figure this out.
Did you get a chance to do the transmission study in ferrets? Your cup of tea, right?
Yeah, my cup of tea. So we're doing it now, and actually we're
Uh-huh.
doing it with Jessica Belser,
Uh-huh.
with ocular inoculated ferrets. So she's doing it in immunologically naive ferrets, and we've sent her some H1 immune ferrets to say how does it change when there's H1 immunity. But it's
Okay.
only direct contact because this virus does not have airborne transmission potential, at
Mm-hmm.
least that we've seen in ferrets so far.
Okay. Are there any transmission from cattle to other animals?
Yeah, definitely cats. So on these farms, one of the big things was cats will drink the spoiled milk, or be in the milking area and drink any of the milk that's sort of like on residual fomites. So you talked before about like other surfaces that are contaminated, and the cats are definitely getting sick. So there's been a number of reports of domestic cats on these farms. Also, phenotypically, there have been, if you look at phylogenetics, you'll see spillovers from cows back into birds because there's a couple mutations that were unique in the cows that have emerged, that have gone back into birds.
Okay. Seema, the cow get really sick after infect. You describe the symptom?
Yeah, so definitely there's been a few cattle that have died or been sacrificed because of,
Oh,
กรพันธุ์
sort
นั
of
กวิชา
clinical
การ
signs,
คอม
but
พิวเตอ
not
ร์ชำนาญ
many.
การซึ่งเป็นผู้ที่มีความรู้ความสามารถ
I'm
ด้านเทค
sorry,
นิคสาร
Seema.
สนเท
That's
ศ
okay.
อีกทั้งมีประสบการณ์ในการพัฒนาและ
Okay. Okay,
Yeah.
sorry,
And
Seema.
no, it's fine. And so what we know is that they're not getting severely ill. So many of the cows that do test positive are not severely sick, but there's like two critical clinical symptoms that seem to be coming out. One is that they have a lower production of milk. So there's, at least in the US, there's so much science behind dairy cows that I had no idea. So every farmer knows how much milk a cow is expected to produce at whatever time they are in their lactating cycle because they have so much data on that cow and on these curves. And so they know when the cow is underproducing. And so underproducing cows tend to have have the virus. So that's one of the symptoms or signs. And then the other one is rumination, like a lack of wanting to eat. And so some of the cows that are dying from lack of appetite and emaciation. They're just not eating enough. But those are two of the more prominent signs that we've seen, at least it's been
I
reported.
Yeah, I remember you say something like the milk turned to be kind of like yogurt or something like that. Is
Yeah.
that gonna be something that the farmer kind of like keep their eye on it if something
Yeah.
happens?
Yeah, exactly. So the forage shipping process, the farmers are looking specifically for that because that milk can't go into the milk supply because
Mhm.
otherwise during testing it'll get knocked out, and it can ruin the whole milk batch. And so that is definitely screened for. However, that might be a viral load issue. It's unclear whether every cow because we're definitely seeing milk virus in milk that looks normal.
Mhm.
Meaning like it's been reported by many groups that the milk can look normal and still have infectious virus in it.
Okay. So next one, did you find IgA against H1N1 in milk or in the cow nasal discharge?
Yeah, so people are looking at serology of cows now. I have not seen any good data yet on antibodies in milk or in the cows. It's a challenge, I think, but people are trying to figure out to detect it because then that's a quick way of saying, did this curd or this cow have H5 if we're no longer detecting the virus? Yeah,
Mhm.
absolutely.
Seema, I think this is the following question. Any seroprevalence data in the no?
Yeah, no, it's really surprising. This has been something that is but apparently a real challenge. So because these are commercial farms, the government agencies just can't go on there and say, I'm going to bleed all your cows. And so it's been a real challenge to get farmers and partners to like allow public health agencies to go in and do seroprevalence studies. I think they're making inroads now, like the USDA has been working really hard to do that, along with like state public health agencies, but the USDA itself does not have control over these farms. The CDC can't go in, the USDA can't go in. These are private businesses.
What about in the farmer, the human that work in the farm? Are there any serology data come out from those population?
Not yet. So the people that have been infected I think sera was taken, I believe, but I haven't seen any reports, nor was there any follow-up. And it really is that there's like, it's a perfect storm, really, Cope, right? The people that work on these dairy farms tend to be migrant workers, so they're undocumented. And so they don't want to seek any medical attention. They don't want their name reported in anything. They don't want to go for follow-up visits. They are like, they're like, they don't want to talk to anybody with a clipboard or a white coat.
Yeah.
And so it's a real struggle to get consent from people to say like, oh, we won't say anything. We just want blinded samples, but nothing. I think it's
Okay.
been I haven't seen any good data yet on that.
Okay, but you are planning to do that? No.
There are many groups planning to do this. Yeah, the CDC is trying to set something up. I know that they've been working on this, but it takes a lot of work with the state public health agency. So again, like it's sort of a weird thing about the US, right? The US federal agencies can't come in and tell a private business how to run their business in a state. Only the
Mhm.
state public health agencies can do that, unless that state is doing something that's crossing state boundaries, right? So this is why the USDA could say, if you're going to move your cows from Texas to Georgia, I can look at your cows. But if you're moving them within Texas, I have no say. Only Texas
Oh,
can tell you whether you can or cannot move that cow. And only Texas can really say like, okay, I'm going to give you enough incentives to make you, want to, help us out and enroll your people in these studies or have the connections. I mean, it's a really interesting space we're in.
because from the map that you show, it seems like many spot that is very far away, but you get some kind of like positive cases.
Oh my
How can
gosh,
that happen? Yeah.
yeah, I had no idea, but we have in the US apparently every week about 50,000 cows are moved from one place to another. And there's no government agency tracking where cows are going
Oh.
for this transport. Like cows can be going from Texas to Idaho so because this process is complicated. So these lactating cows get milked, and then they go to what's the dry period, and then they get re-inseminated. And so when they're re-inseminated, they maybe go from, okay, say they were milked in Idaho, and now they're going to go down to Texas to get inseminated and eat because the land is cheaper in Texas for them to feed on. And now from there, they're going to get moved. Once they have started the process of lactating, they get moved to now North Carolina, right? It's like the chains are just so all over the place.
Okay. Seema, I think this is important question.
Yeah.
After recover from the infection, how long that milk is safe for consuming?
Yeah, so pasteurization is definitely killing the virus. I mean, based on the data we've seen so far, pasteurization is killing the virus. And so any pasteurized milk is safe to consume even now. And it is raw unpasteurized milk is never safe to consume, at least, because of bacteria as well. And so I think the experimental inoculation studies that are being done by, the USDA, Amy Baker, and Andy Bowman, and others are really going to tell us how long animals shed virus in their milk. And from my understanding, it's pretty long.
Okay. So, Seema, can we go back to the basic virology question? One more question.
Yeah.
Do you know the receptor distribution in the cow?
Yeah, so cows, I think people have recently started looking at this. We didn't really know, right? Because it was only influenza D. It's really been reported in cows, but it seems to be that cows in the mammary gland definitely have 2,3 as well as 2,6 sialic acids. But the distribution, the relative amounts, the branching patterns, all of that is not, I think, well understood yet.
Okay, I think this is some other question about the clinical side in bull. Seema, can you describe a little bit?
I don't. So there's been in the bulls, there's only been a couple of ones that are non-lactating have been observed, and I don't think we have a good handle on the clinical signs yet.
Okay. Are there any other question from the audience? You can raise your hand and turn on your microphone to ask as well. I think one more question here. Can the infected milk transmit to their calf and act as a reservoir?
Yeah, that's a good question. And definitely feeding contaminated milk to, like, say, mice that Yoshi did does infect the calves, infect the mice. Most of on the farms, at least that I have been to or that I have seen in these commercial industries, the calves and the moms are separated almost immediately after birth. And so the calves don't necessarily drink from the mom. And so they're given something separate to initially get them off the colostrum and other things, and they're bottle fed.
And Seema, when you work with people in the farm, usually when they get infected cow, they separate those cow from the other. Is that right? So prevent,
when they have a sick looking cow, it's separated, but I don't know how often the clinical signs manifest to that level.
Okay. Are there any other questions? You get tired, huh? It's getting late over there.
Getting late, yeah.
Okay. I think if there are no more questions, I would invite either Ajahn Prasad or Ajahn Pilaipan say something to thank Seema for joining us today. Ajahn Prasad, please. I think Ajahn Pilaipan.
No, Ajahn Prasad.
Ajahn Prasad,ค่ะ. He is the vice president of the Virology Association Thailand. ค่ะอาจารย์
Yeah, good evening. I came to join a little bit late, but this is a very informative and really interesting lecture. On behalf of the Virology Association and the Virology in Infectious Disease community in Thailand, so to say, because these are first-hand information that we don't get quite often. We would like to thank you for spending time and know that it's late there. We appreciate your time. Thank you very much again. Thank you.
ค่ะ thank you ค่ะอาจารย์
Thank
ประเสริฐ
you.
Seema, thank you very much. Definitely invite you again next time for the other talk. And I think it's getting late over there. I let you take a break and take some rest for tomorrow. And I would like to thank all audience in this channel that join us. And hopefully we meet you next time with more interesting topic. All right. Thank you
Yep.
very much. Have a great day.
Thank you so much, Kobe. Bye. I'll talk to you later.
Bye.
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 Thank you.